That being said, today we are going to be talking about DNA. Yesterday you got some vocabulary. You didn't get all of the vocabulary. You will need to get most of it on your time. There are two pieces of the vocabulary that I do want to make sure that you have, because as we go through, these things are going to be popping up. So if you take your vocabulary sheet out, yesterday I said don't worry about promoter, and I said that central dogma was not on there. Today you are going to go ahead, we're going to go ahead and get that. Stop talking while I'm talking. It's very rude. And get that hoodie off because it's not dress code. This page number 45. Right now you're displaying characteristics of a kindergartner because you're yelling out at me. All right. Promoter. That's this word here. What is a promoter? In your own words. Again, today will not be the day that I'm doing all the talking and you're not doing any participating. You're just looking at me, waiting for me answers. No. But not thinking about science, what is promoter? Shania? Something that enhances something? Kind of.
You said outside of science?
Just the word promoter, period.
Like an endorser?
An endorser. Alex, you had your hand up?
It puts somebody out.
Put somebody out there. Builds up something. Okay, so what if I said a party promoter? What's that? What do you mean by promote? You can't tell me the word. Okay, so do they initiate it? Do they start it or do they end it? They started. So here, a promoter is a starter or an initiator. So it initiates, I-N-I. Yes. I-N-I-T-I-A-T-E-S. Initiates or starts. Initiates or starts. Initiate. Don't tell him just put. He wants to build his vocabulary. I-N-I-T-I-A-T-E-S. Your head shouldn't be so big.
No, I'm just saying, can I?
What do you say? Your head was too big. You couldn't get your shirt over your head.
I said, can I keep it around my head?
Can I tighten it around? Yes, you keep your hoodie like that. Just don't put it all the way on. All right. So a promoter initiates. It initiates something. Now, look back through your vocabulary. There were some words that kept popping up within the definition. What were some words that you kept seeing throughout your DNA vocabulary? Shamaika. You had your hand up, and then I called her. She said, dang. RNA. Bases. What else? Polymer. DNA. Okay, what did RNA have to do with DNA? Look in your definition. Kyle.
It's found in the nucleus.
Okay, they both are found in the nucleus, but what do they have to do with one another? Louis? Shh. Huh? Yeah. Because this whole topic is about genetics, which is about DNA. RNA transports information from DNA? Okay, so is it like copying it? Okay, so in order to copy something, if I said, well, if you came to school and I was your peer and you said, hey, Wiggs, I didn't get the notes, I would say, oh, here's my copy. Go ahead and copy it. That would require you to do what? Pick up your pencil and write it down, which is another word for transcribing or transcription. So a promoter initiates transcription, which is the copying of something. And we're going to be talking about that more later. But promoter initiates transcription, which is the transfer of writing. Script means writing. Transcription. You don't need to know what, well, you did need to know what that is because that was one of the definitions as well. Central dogma. This is not the middle of a mother dog. That was a joke. Thank you, Renisha, for laughing because I'm hilarious. All right, central dogma is actually a genetic theory. Central dogma is a genetic theory, Vernon, transcribed right now. Don't give me that look. It's a genetic theory. A theory is something that has been tested over and over again and is basically the same thing keeps happening, so they come up with this theory. Like my theory is that you guys don't like homework. It's been tested over and over again and you don't like doing it. All right. A genetic theory that genetic information is coded, C-O-D-E-D, coded in self-replicating. So that means duplicating itself. In self-replicating DNA. So it's saying all the information is within the DNA that duplicates itself. And protein synthesis. And protein synthesis. What do I mean by protein synthesis? There's that protein again that we've been talking about forever. What's protein synthesis mean? Nope. What does synthesis mean? To synthesize something. Say it louder. Make. So what's protein synthesis? To make proteins. And that's what we're going to be talking about because that is the goal of the cell. Remember we said that. So we're going to be talking about that a little bit more. But before we do that, we actually need to see what this DNA stuff is all about. So go ahead and put your note sheet in front of you titled Modeling DNA or DNA Notes. Before we move on from there, I do want you to recognize that you do have homework on today. It is going to be, see what my theory, you don't like homework, I know. DNA, it's necessary. DNA analysis questions. There's only 5 questions that you'll need to answer. You'll be doing it from your notes. It is on Canvas. It is due tomorrow morning. There is a hard copy in case your internet goes down, your computer breaks, you eat your computer, or whatever you do with your computers when you get home. All right. So, with your notes, DNA stands for deoxyribonucleic acid. Hey, all of you can say it if you slow down and use your vowels. Nucleic acid. All right. With that being said, there are some words within that long word that you can break down. What are some of the words that you see in there? Oxy, which comes from, so it must contain some type of oxygen. What else? Nucleic, so it must be contain what? The nucleus. It must have something to do with the nucleus. Alex says ribo, so what is that for? Ribosome, so it must have something to do with that, and it is an acid. Any questions? Okay. Then on your notes, you should have filled in the part that DNA stands for deoxyribonucleic acid. Moving on. DNA is the blank of all living things. You could possibly fill in this blank. We talked about this on yesterday. You did the vocabulary. DNA is the what of living things? Foundation. Somebody says canvas. What are some other words? Blueprint. We can definitely use, in one of those blanks, put the word blueprint. In the second blank, we want to say genetic material. So even though it's one big blank, you can put both of those words in one blank and put blueprint in the other because those are very important. It is the genetic material, but it's also the blueprint. Like proteins. Remember when we watched that funny little video with that guy with the glasses and the funny teeth, and he talked about biology, and he talked about carbo being a thought and all that other good stuff, carbon, remember? And we watched the video and it talked about the 4 essential things for life. And you guys were telling me you need water, shelter, and all that stuff. And I said, yes, you do need those things, but the major 4 essential things was protein, which we talked in depth about. We also talked in depth about carbohydrates, and we talked in depth about lipids. And I said the 4th one was nucleotides, which we're going to talk about later. Later is now. But before we move on and talk about those nucleotides, DNA is a polymer. And Louis said he recognized that word from yesterday's vocab. The definition for polymer is right there. It's a large molecule made of many repeating subunits. Those subunits are monomers. God bless you. The subunits of DNA is that The subunits of DNA is that 4th thing that is essential for life, which are nucleotides. Then on your notes, it tells you that DNA contains information that codes for life processes. Life processes is everything that you do in life. The major life processes is taking in what? Which are considered, starts with an N, nutrients and getting rid of waste. That's the major life process, and your DNA codes for the information so that you can do all of those things. But understanding this chemical structure of DNA, deoxyribonucleic acid, allows the scientists to understand how DNA codes for specific things. And the major goal of a cell is to do what? Make something. What is it? One of the 4 things, what? Protein. So understanding how DNA is structured helps scientists understand the codes for protein. Protein is the word that you're filling in after DNA codes for proteins. Proteins are essential and necessary for our life. We recognize that when we study proteins. Scientists also need to understand, and we don't need to write anything just yet, but scientists need to understand the structures of the DNA for a couple of reasons. We want to be able to understand the desirable genes and the non-desirable genes. Desirable genes would be things such as, why are people geniuses? Why are people good in math? Why are people good in reading? We want to see, is there something genetically within them that helps them to be good at these things, and can we copy those to give it to somebody else? Also, with genetic diseases, we want to be able to see what is in their genes that cause these diseases, and can we get rid of it? Understand? As we go through this activity, you are going to model a structure of DNA, and this is going to help you get some information about how DNA is structured and how it's put together, and understand it a little bit more. All right. So, on your notes, DNA is what chromosomes and genes are made of. What chromosomes and genes are made of. Now, before you start filling in any of your other blanks, just wait so that I can break it down so you can understand it as I go. If you're writing while I'm talking, chances are you're not really paying attention. So this picture explains exactly what's going on. How many of you have ever had a piece of string hanging from your sweater or your jeans or your shorts or some type of article of clothing, and you just go to pull it thinking you're going to rip it off, and you start pulling the whole thing apart? It starts unraveling, and you now see that your sweater is made up of much more than what you thought it was made up of. Okay, that is essentially what we're looking at here. This portion here is a what? No. This is a cell. So on your line that you have there, you want to label this a cell. So we're looking at the cell. Because again, we're still talking about cellular biology. So we're looking at our cells to help us understand. Now remember, the cell is very, very, very small. And all of this information is within a very, very, very small cell. Your cells are very important because they contain a lot of stuff. Over to the left side of your notes, it tells you that chromosomes contain DNA. That's why it says DNA is what chromosomes are made of. Chromosomes contain DNA, which is made up of these subunits, these smaller units. Those subunits are the 4th essential thing to life, which is nucleotides. You wrote it once, you're writing it again. You don't need to see how it's spelled because you already wrote it. This word is nucleotides. Bubakar, you wrote it already. Copy it from that. Sit down. You shouldn't be. The only thing you should be looking at is what I'm talking about right now. I know. You won't get the last word. Guarantee. Stop. All right. Moving over to the nucleus now that we've talked about these chromosomes that contain DNA, we need to now take a closer look into the cell. So we look deeper into the cell, and the brains of the cell is called the louder nucleus. If you can't see this word on your paper, you want to go ahead and label that nucleus. I know it's kind of fuzzy on your paper. All right, and then I can do this. 1 second. Then when we look here, you notice that within the nucleus are these little X's. They kind of look like a bunch of little X's. These X's are called chromosomes.
There we go.
All these chromosomes, which contain your genetic information, which tells you who you are, why you walk the way you walk, why you talk the way you talk, why you blink the way you blink, why your eyes are shaped the way they are, why your hair color the way it is, why your skin is like it is. All of these things are within your DNA, within these little X's of these little chromosomes that are there when you are being who you really are. These chromosomes break apart and duplicate themselves to recreate cells, because your cells are creating all of the time, and when they break apart, the sister chromosome is called a chromatid. That's why those words are under there. The center portion is where the chromosomes split and break to create new chromosomes, and this is called, and when I say new chromosomes, I mean it duplicates themselves. Can't do that. It's hard when you're not actually writing on a smartboard. Yeah. But that is a centromere, centromer, centromere. I'm trying to enlarge it. There you go. Oh, no, large too much. Centromere, centromere. That is the center of the chromosome. And that's where it's going to split and break to recreate when we talk about mitosis. This is what we're talking about when we say chromosomes duplicate themselves. So this now gets into that portion where you unravel your sweater. Within these little X's, they're raveled up, kind of like a phone cord. When your phone, those bungee cords that Apple has now made, where you pull them apart. But when you pull them all apart and you unravel them like a sweater, then you will realize within that unraveling, you have protein. Yeah, it won't let me. Then, when you look within those proteins that have been made, what may help make the protein, or where the protein is located, the unraveling of that, you then have DNA. Nope, won't let me do it. Why aren't you writing? Okay. The words under DNA is double helix. A double helix is a twisted ladder. Like taking a ladder and then twisting it. Shamika, stay with me and you will hear everything that I'm saying. Just stay with me. Because right now you're disrupting somebody else. Looking within the DNA, remember at the very beginning, I just told you that your DNA is what chromosomes and genes are made of. So within your DNA, nope, you have your genes here. And these aren't the genes that you put on Fridays or on the weekends. It's GENE. And that again tells you your color, your hair, the way you act, the way you walk, the way you talk, how deep or not deep your voice is, all of that good stuff. And then your DNA contains these base pairs, which I wish I could make a perfect square. Nope. Base pairs, B-A-S-E pairs, not the type of base like Kanye's shirts with the holes in it. I hate them. I don't hate Kanye, I hate the holy shirts. I know that's crazy. Wait, let's not get off track. Kyle, what'd you say? Nope, nope, it doesn't change when you mature, when you start to develop these things. All right, moving on. Shh. Now we have our picture and we understand where DNA comes from that's located in the cell and how all of that is made up so that we have a basic understanding of it. We're going to go ahead and move on a little bit further. DNA is a repeating subunit. It's called what? Nucleotides. Please make sure you fill in your blanks. Now breaking down a nucleotide. A nucleotide is made of a, nope, look up at the screen. Don't say you don't know. Sugar, write it. A phosphate, you don't need to put phosphate group. You can just put a phosphate and a base. So nucleotides are made up of a sugar, phosphate. Sugar, nucleotides are made up of a
Sugar, phosphate, base.
Say it again.
Sugar, phosphate.
Very good. In your bag, you're going to identify each parts of these. There's one bag for every 2 people. Before you move on, let me just show you something here. You should, quiet, pay attention because we need to get all of this done. You should have one big bag that looks like this. There's going to be a Ziploc bag that has a big purple half circle. Take that and just put that to the side for a second. Then there's another Ziploc bag with some purple and some black pieces. Put that in front of you because you need that. In the baggie that has the little green, purple, yellow, orange, and white pieces in there, right now take out a white tube and a blue tube that looks like this. Yes, you do. All right. Sit here. Here you go. All right. Jonte and Alex. That's the last time I'm going to talk to you, and I'm on camera now telling you that. All righty. So now, in your bag, you're going to take these pieces out and you're going to identify them. So just put them in front of you. Red? There's no red. White, blue, and? I'm sorry. What colors are we taking out? White, blue, and? Look at the screen, young people. What color? White, blue, and black. This, I said, first of all, I said we're going to take out the pieces like that are on the screen. So therefore, when I started pointing out the bag to you, I pointed out to you what the bags have. Then you look at the screen and you notice the pieces that I have look exactly like the pieces that are on the screen. So you need this little black piece out, along with the blue piece, along with the white piece. They look just like the picture on the screen, guys. Stop making this more difficult than what it needs to be. Now let's stop talking because we need to get this done. Now the nucleotide, all of this is a nucleotide. Why is it a nucleotide? Because it's made up of a sugar, phosphate, and base. All right. So when we look at this, the little white tube, get this over. The little white tube, I do both. The little white tube is the what? Phosphate. The black piece is the what? Sugar. And the blue little tube is the base. So far, so good, right? All right. So when we think about DNA, we think of it like a ladder. On the ladder, you have sides that you have to hold on to go up the ladder, correct? In the middle, you have rungs or steps, correct? All right. So when thinking about DNA, the rails, the sides, are here, and the steps are here. One part of the rail is the white part, which is a phosphate. Make sure you fill that in. Another strand or rail of the ladder is a sugar, and here we're going to call it a deoxyribose sugar. And we saw ribose in our ATP. The middle part, or the steps, are the bases, and these are nitrogenous bases. So again, our nucleotide is made up of phosphate, sugar, and base. So far, so good. Okay. Now get your color pencils in front of you. And you may need to share some colors. Let's not talk while we do this because we really need to move. Now, there are 4 types of nitrogenous bases. So there's 4 bases. These are the parts that make up the middle, the steps. One base is adenine, which is A. In this demonstration, adenine is going to be orange. So next to the A, put a little orange. Dot or line, whatever you choose. So A stands for adenine. Everybody say adenine. Adenine. These are not going to go in order of your notes, so pay attention. C stands for cytosine. Everybody say cytosine. Cytosine. Next to the C, write the word cytosine and put a little blue dot or a blue line next to it. Use the purplish blue. All right. The third type of base is guanine. Everybody say guanine. Guanine. Guanine is going to be represented with the yellow tube. So put a little yellow mark next to it, or write the word yellow if it doesn't have a yellow in your color pencil box. All right, guys, there's no reason why we can't finish. The fourth type of base is thymine. Everybody say thymine. Thymine. Everybody. Thymine. Shania, you have not said it yet. Waiting for you. Thymine. These are not future names for your children. Put a little green line or a green dot next to it. I don't want to say, come here, little adenine, little cytosine. It's Shemaiah's daughter named Cytosine. Third event. All right. All right. So again, how many bases are there? 4. A, C, G, T. Adenine, cytosine, guanine, thymine. All right? Are we good? All right, moving on then. Now, this says with your partner, but you're not going to, well, actually, you are going to do it with your partner because some of you are working with the partner. I'm just trying to make this so that you all can see it on the screen. All right. With your partner, you are going to use the DNA model kit to construct the DNA nucleotide as shown in the above figure. Remember, a nucleotide consists of a phosphate, sugar, and a base. So you are going to, with your partner, create this right here. No, you're going to put it together from the materials in the little baggie that you just took out. It shouldn't take us more than 10 seconds, 10th graders. 9, 8, 7, 6, 5, 4, 3, 2, no ma'am, 1. All right. You should have something that looks like this. You have the white part, which is the black part, which is the, and the blue part, which is the, which base is this? No, which base is it? You put a color code next to yours just a moment ago. No, sound it out. It's your future daughter's name, cytosine. Well, you just put blue next to it on your chart. And this is blue. Yeah, this is the base. There's 4 bases. All right, stay with me. Everybody with me? Stop talking over here, please. All right, now you and your partner, let me get this where you can see it. You and your partner are going to continue to build and connect a single chain of 4 nucleotides using any sequence bases. Now, before you do that, let me just explain something here. Notice in here, this is 1 what? Nucleotide. This is 2 nucleotides. This is 3. How many are you putting together? 4. It says that you can use any sequence of bases. What are the 4 bases? Adenine, guanine, cytosine, thymine. There are 4 colors: orange, yellow, blue, and green. You're going to put them together with any sequence of bases. If you notice, in your baggie, you have orange, you have yellow, and you have green. And then there's also the blue one. We're not doing anything with the purple one. These are the orange ones. It's like a tan color. So now go ahead and put together 4 nucleotides. Huh? No, 3 more. You got to put together 3 more and put it together because you already made 1. Use whatever order you want to use. Whatever you don't use, just give it back to me. Again, you and your partner together. What? You tell me, Shamaika. This is not rocket science. It's biology. I don't know. You put it together. You tell me. I know. It's so hard. It's so hard just following directions. I know, but you do follow them, but it's hard for you. Oh, it's just difficult. You need another. You're putting it together incorrectly. These should be what color? Those are your phosphates here. Here. Yep, only 4. 5. 4. 3. 2. 1. You should be done. You should have created something that looks similar to this. Now maybe your bases may look a little different because maybe you've chosen a different sequence of bases. But you should have phosphate and sugars being the ladder, the handle of the ladder, correct? And the bases are the steps. And we have 4 nucleotides. We have a phosphate, a sugar, a base. Phosphate, a sugar, a base. A phosphate, a sugar, a base. A phosphate, a sugar, a base. Does that make sense? All right, put it down in front of you for a moment. In the mid-1940s, a scientist by the name of Erwin Chargaff analyzed the percentages of each four nitrogenous base. These are the bases that we're looking at on here. He analyzed it, and when he did that, he summarized the following information below. He noticed that when he looked at the bases within humans, he looked at the percentages of each base within the human. He looked at the percentages of each base within the rats, within a sea urchin, and within a salmon. He looked at these different percentages. Right now, I want you to look at this chart. Make it smaller for you to see. And discuss with your partner the patterns that might be suggested about the nucleotides in DNA. Again, the key word is patterns. Take a moment and discuss it with your partner. Take about 3 minutes. You should be looking up at the chart.
Oh, I like it. I like it.
1 minute. All right. Up here. Ladies, have a seat for a moment. All right. Over here. Tell me, what did you notice about a pattern at all? Over here. And that's not telling me anything about the pattern in between. We're not looking at a pattern between humans and rats and humans and sea urchin and human and salmons. We're looking at the pattern of the bases within that particular species. Around the same? For humans, it is the same. For, what about adenine and cytosine? Adenine and thymine. What about them, though? What about their percentages? What about the percentage of adenine and thymine? They're very similar to each other. What about those same things in rats? Is that same statement true in rats? Is the statement true in sea urchin? Is the statement true in salmon? Yes. So that's not my issue, honey. So based on the conclusion of seeing that within species, adenine and thymine are about the same percentage, which means that the same amount of adenine in a human equals the same amount of thymine in a human. The same amount of cytosine in a human equals the same amount of, and the same thing for all species. Is that safe to say?
Yes.
So, using that information, we're going to construct a complementary strand. What does complementary mean? Let me say this. Jay-Z compliments Beyonce. Kanye compliments Kim Kardashian. They go together. You don't see one without seeing the others, or most times they go together. Is that correct? So, looking at this chart, which base would you say goes with adenine? Thymine. Which base goes with cytosine? Guanine. Because what it's saying is, humans, we don't have just one strand of DNA. We have two strands. So if adenine is 30.2% and thymine is also 30.2%, it must mean that however many adenine we have equals the same amount of thymine we have. So that must be the matching pair. Do we understand that? So now it says, based on your conclusion, we're going to construct a matching strand. So let's take a look here. Before we do this, on your notes, it says bases fit like a puzzle, pairing in the middle of the strands. They're going to fit together. So A stands for adenine, binds to what?
T.
Which is thymine. Put that T there. It's only going to bind with T. That's the only thing it'll fit with. What binds with G only? C. Cytosine. So right now, we are going to go ahead and create our second strand, putting it together. But when we do that, let me explain to you how we do it. First off, looking up at the screen, my first base is orange. So that is what? That's adenine. So what is going to complement adenine? Thymine. Notice those two letters both have straight lines. A has a straight line, T has a straight line. So when we combine them together, there's another little white piece in your bag which has a rounded top and a rounded bottom. You're going to put that in the base that you're going to be connecting to, and you're going to match its base pair. So again, I take that little white piece so that I can connect adenine with thymine. But look up here before you do it. This is not a finished nucleotide. All I have is a base. What else do I need? All I have is a base. What else do I need? A sugar and a phosphate. So remember, my base is connected to the phosphate. I'm sorry, to the sugar. And then I have the phosphate. So I did my first rung. You finish and put your complements with the other. Do that now. Here. Let me just get out what I need to take this back so I don't want to lose it. It should take you no more than 2 minutes to do this. Oh, you need these too. Here, Renisha. Oh, Renisha, I need some white tubes. One minute. Thank you, Asia. All right, you should have come up with something that looks like this. Notice orange, which stands for adenine, always goes with T. Thymine green. Thymine goes with adenine. Here you have yellow, which is guanine, going with cytosine. Cytosine going with guanine. I already went over that, Shamika. So the middle portion is your base pair. These are your base pairs. We're going to skip the drawing for now because we don't have time, but we're going to pick up on the drawing on tomorrow. But if I can see someone else's that's already together so I can show you something, bring it here, Terrence, since you're closer.
Here you go.
And Asia, bring me yours. When we put it together, this is the bottom portion of your notes. I have a lot of talking over here, and I don't like it at all.
Good afternoon, Wildcats. We hope you all have had a wonderful
We
day.
still got 3 minutes.
We need all beauty club
put it
members
together.
to make sure on your way out at dismissal
We
that
noticed
you
that it
stop
forms
by
a
the
what?
front desk and get
A double
card
helix
from Ms. McCall.
of
All
DNA.
students that are in the beauty club, remember we do have clubs
So
tomorrow.
looking at
All
your
students
note
that are in
sheet,
the beauty club,
DNA
as you are
has
exiting the
a
building at dismissal,
double
please
helix
make sure that you
structure,
stop by the front desk and see Ms. McCall to get
also known
information on
as a
that
winding
must-see ball back
staircase. Fill in your
tomorrow.
bottom
You
portion of
must
your
bring it back
notes.
tomorrow to participate in the activity that you guys are doing in beauty club tomorrow.
Right there. DNA has
Also,
a
dance
double
team, please remember
helix,
there is practice tomorrow until
which is also known as a
5:30.
winding
Please make sure your
staircase.
ride is here to pick you up at 5:30. All dance team members, make sure your ride is here to pick you up no later than 5:30.